All right. My name is Brett LeVay-Young, and I am right now the head of sales for libraries at DNA 2.0. If you ever request information about a library or a quote for a library, you'll be directed to me, and we'll be communicating at that point on the specifics of your project. I'm going to give people another couple of minutes to join us if they're joining us late. And what we're going to cover in this webinar are mostly basics on discussing libraries and ordering libraries from DNA 2.0, getting information about libraries should you have not ever used those in the past. So if I go to my first slide here and talk about what variant libraries are, for the most part we think of them as tools for examining and improving proteins that you may be using for whatever function you have, whether that's antibody libraries, enzymes where you're trying to improve various properties. Whatever you have, you can potentially examine the functions of those proteins using variant libraries. And if you have a protein where you know very little about the function or how to improve it, you can actually use libraries to examine that as well. Libraries are going to come to you either as a collection of individually sequenced genes or pools of multiple genes. And the second, the pools of multiple genes, tend to be the kinds of things that most people think of as a library. You can use them to improve the properties of antibodies, target binding, get either higher affinity or specificity, or even to change the specificity of an antibody. For enzymes, to increase the activity, the thermal or pH stability, substrate specificity, and any number of other properties. You can use the libraries to also try to increase the solubility of a protein, and I know that crystallographers are particularly interested in that function. On this slide are also a number of customers who have purchased libraries from DNA 2.0 and or published using those libraries. Now at DNA 2.0 we have two basic ways of going after changing the function of proteins. The first is libraries, which is what we're talking about today, and we tend to think of libraries as having a relatively low input in terms of design and a much higher input in terms of screening effort. And so essentially we are making, you and we will collaborate to design this library. We'll build it, and then you'll spend quite a lot of time screening the library to improve or select function. We also offer protein GPS, which is our our protein engineering service. And there we spend much more time on the design phase of the project with the goal of offering you a smallish number of genes on the order of 100 or so that you can then apply to a screen that may be much more meaningful in an informational sense. We are, like I say, going to focus on libraries here today. So the first library type is an alanine scanning library, and this can be used to determine which residues of the protein are critical for some protein function, whatever that function is. And so if you look at this diagram here, you have on the top a protein sequence. And what we're going to send you if you order an alanine scanning library or a leucine scanning library, any scanning library, is a collection of genes individually sequence verified and shipped typically in a plate, where we replace one residue in each gene with an alanine, a leucine, whatever you specify. Many customers seem to think that these, because they're conceptually simple, should be quite inexpensive. Unfortunately, that's usually not the case because we're shipping you individually sequence verified genes, and that increases the cost of the library. And you can see that the delivery format is typically enumerated sequence verified clones. And the goal for this library is to look at the properties of each individual protein that's derived from the gene and look for specific mutations that typically are going to knock down, knock out, or decrease the function that you're examining in that assay. This is not something that's typically going to be used to improve the function of a library, although I suspect that in some cases that might actually happen. The second type of library, and this is something that we more typically ship to customers, is a so-called site scanning library. where you are going to take your protein of interest, determine an individual amino acid residue, in this case a glycine, that you want to examine, and you may have found that perhaps by using an alanine scanning library or structural information. And you're going to replace that residue with a variety of other amino acids using some sort of ambiguity. And then we're going to specify this here as an NNN, which is going to give you all 64 codons, including stop codons, as part of what you receive. You can receive these either as a linear DNA fragment, which is just going to be sequence verified on that, as a ligation product in your vector, or even as enumerated clones. That increases your cost, but also gives you something that could be much more useful functionally. And the goal here is to see if by substituting an individual amino acid into a single position, you can improve the function of a protein. You can then later decide you maybe want to combine several of those mutations into a single gene. One of the important factors in looking at a site scanning library like this is how even the diversity that you receive is going to be in any single position. And this is quite a large concern for a customer that we had who was receiving libraries from another company and was not getting an even distribution. And so they took some libraries that we sent them, cloned them, and sequenced 10,000 or 5,000 different clones using high throughput sequencing. And they determined that the libraries they received from us have an extremely even distribution of nucleotides in the ambiguous position. And this is exactly what they were looking for, and they've now gone on to order a number of other libraries. They typically order these between 50 and 60 at a time to examine 50 to 60 different positions in the proteins that they're trying to improve. So the important point is that not only is the distribution even, it's also independent of the location within the ambiguity position itself, the location within the gene, and the preceding or subsequent nucleotide in any of the single positions that they examined in this particular case. The next type of library we're going to talk about is a combinatorial site library, and this, I think, makes up the bulk of the libraries that customers tend to order from us. And this is, in fact, the kind of thing that many people almost always restrict the idea of libraries to. So, in this case, you have your protein, and you've now picked several residues, in this case 4, that you want to diversify in a specific pattern. And of course, if you choose, you can diversify these using the same ambiguity that you saw before, the NNN or the NNK. You can also call out specific amino acids, specific codons that you want to substitute as the pattern here, where in some cases you're only substituting in 3 different amino acids or codons, and in other cases you're using large numbers up to 18 to 20. The diversity that you receive in a library like this can rapidly grow to very large numbers, and that's something that you want to pay attention to as you think about doing the designs for these libraries. We try to encourage customers, when they're ordering this sort of a library, to restrict the diversity to under 1 million at a time. But we have had customers that order very large diversity and get good results with that as well. You can receive these as linear DNA fragments again, ligation products, into your own vector, or as enumerated clones. Of course, with enumerated clones, if you specify a diversity of 10 million, we're not going to be able to deliver that as enumerated clones. And of course, we'll discuss that with you during the order specification process. Now, one of the factors in thinking about a site or a library like this is that in addition to simply specifying a selection of codons in any single position, you can also think about trying to bias the distribution of codons within any individual position. This slide, which actually has way too much data on it, is an example of a situation where customers specified in some positions, you can see here, whatever the specification is for this particular color of blue, they wanted a lot of that codon in this position with a, if you will, a sprinkling of other codons. And in other positions, they wanted essentially an even distribution of a large number of different codons. So this shows you that the predicted distribution and the actual distribution that we found after sequencing almost 270 individual clones are relatively similar. Statistically, I don't think that you would be able to determine that these are different from each other, the predicted versus the actual, and that we managed to match, for the most part, the customer preference in terms of codon distribution, and with relatively few codons that the customer did not, in fact, specify for any individual position. And of course, no one can tell you that they're going to match anything perfectly and that there's going to be no errors. You'll see here that for this particular library that we made for Antibody, 22% of the genes that they pulled out, or that we pulled out and sequenced, had non-design mutations. And in this kind of a library, most of those non-design mutations are going to end up being frame shifts. So they, with any luck at all, will completely fall out of your screen. Now our guarantee specification for this kind of a library is typically going to be that you will receive no more, or a library with no more than 40% of the clones showing non-design mutations. Whenever we do these specifications for you, we're essentially giving you an upper limit on what you can expect to see. We always try to do much better than that. So while this customer received a specification that they would receive no more than 40% non-design mutations, in fact, they got much closer to 20%, about half of what we predicted. Now, there are limitations on combinatorial site libraries, and this is something that we are going to discuss with you if you make a specification that we look at and see any red flags. We have had customers, for example, specify diversity that is much, much larger than we can meaningfully provide. For example, we had one customer that wanted a library with a diversity that he specified, or she, at 10 to the 57th power. And that would require more molecules than are on the entirety of the Earth. So we'll talk to you about whether we can actually provide for you the specificity that you're trying to get. For example, the example I have here is if you take a gene and you specify 12 NNK positions, NNK then gives you an encoding of 32 different possibilities. The diversity there is 1 times 10 to the 18th, which is really larger than we're ever going to be able to deliver to you at all. And that is, for example, for a 100 base pair library, which is a relatively short library, 1 microgram of DNA, which is our typical delivery specification, is going to be about 10 to the 13th molecules. And that's far, far, far less than 10 to the 18th, obviously. In fact, even for a 100 base pair library with this kind of a delivery amount, even though we are delivering approximately 10 to the 13th molecules, in fact, the actual diversity there is probably going to be less. And we would prefer to think of that as a diversity for this library of 100 base pairs of approximately 10 to the 12th, so an order of magnitude less. So you can see that even if we deliver much more DNA, even if we could, and we're not going to be able to, but if we could deliver a milligram of DNA, you're still only going to get on the order of 10 to the 15th as your diversity, and not 10 to the 18th. If we need to use a type of synthesis called a split pool oligo in order to make the kind of library that you're specifying, that's going to further limit the diversity. And if we see a library that's specified in that way, we're going to talk to you specifically about what the limitations are going to be and what we can possibly deliver for you. And then from there, we can further think about what might be better ways to approach your specific problem. One of the advantages of talking to us at DNA 2.0 about your libraries is that you're not just talking to me because I'm just one guy, and in honesty, I haven't been thinking about libraries for my entire life. You're also talking to the really extremely smart scientists we have here at DNA 2.0 who think about libraries. They must think about libraries in their sleep because they're constantly coming up with new ideas and new ways to make these things that I would never think of. So as you talk to me, I'm going to then try to understand what you want and then transmit that to people in the lab so that even if I think we can't make your library, I'm going to go ahead and talk to people in the lab about it to see if we can come up with some way to give you what you need. One of the issues in combinatorial libraries is actually meaningfully screening the diversity that we send you. Now, this is a graphic which is on our website, and it has quite a long explanation on the website that I'm going to try to simplify here for you. If you find this confusing, you can take a look at the website and read the explanatory material. And if you're still confused, you can give me a call and we can go through it some more if it's of interest. So here what the situation is a library that has a specified diversity of 3 times 10 to the 6th. So a diversity that's not small, but certainly not outrageously large. And the point that this is making is that to meaningfully screen most of the diversity that's going to be present in the library, you're actually going to have to look at something on the order of, excuse me, I'm going to have to back up here and see if I can get rid of this. There we go. Sorry about that. So you're going to have to screen on the order of 10 to the 9th different clones to see the diversity that is present, even though it's only specified at 3 times 10 to the 6th. And that is because thinking about protein sequences, the genetic diversity that you get in something like an NNK specified combinatorial library there is a lot of ambiguity that generates multiple encodings of a single amino acid in a particular position, so that, for example, something like arginine or serine may be much overrepresented in the library. So to see a variant that has something like a tryptophan or a methionine in a specific position, you're going to have to screen many, many more clones than you would ordinarily think if the encoding was unbiased. So one of the things to keep in mind as you think about libraries is that even though you might want a library where the diversity is specified as just, for example, 10 to the 9th, in fact, you may be having to screen 10 to the 12th, 10 to the 15th different clones to actually meaningfully see that library. And we may not be able to provide that much physical diversity in the product that we send you. A newer type of library now that many customers are interested in are so-called modular assemblies or BioBrick type libraries. And we have several customers that are ordering these in large numbers, and it's something that's generating a lot of interest. The goal of this library is to assemble a bunch of individual elements that may include, in this case, promoters, open reading frame genes that encode proteins, individual terminators, and then different vector backbones, and then assemble those in specified formats. To make these actual individual brick elements, so for example, the individual promoters, the individual genes, those are essentially set up just as a standard gene synthesis. We then save these for you in 96-well plates in the freezer. You can then either specify, either initially or later, specific combinations of these different elements that we put together for you and then ship to you either sequence verified, or if you're very comfortable with the process, not verified. Typically these are shipped in 96-well plates because often you will be ordering or thinking about large numbers of these. And the pricing per individual gene can be quite reasonable because we're essentially reusing the same elements and designing them in ways that they will only go together in a single pattern. We can help you with these design elements, and in fact, we may need to revise any designs that you come up with because we have specific requirements to put these together. But we're happy to do that, and again, you're not just talking to me, you're talking to a lot of really smart people here at DNA 2.0 if you decide you want to think about this kind of a library. Now, there are additional libraries that we offer. Their demand for these is somewhat lower, so I'm not going to discuss these in any significant depth. A random library is something that's generated by error-prone PCR. These tend to be relatively reasonably priced. The mutations are completely random. They're going to be mostly substitutions, not truncations or deletions. So they tend to have a higher representation of useful random clones, but the mutations that you're getting are completely random. Like I say, these are reasonably priced, but at least in our view, they're not as useful because there's almost no design that goes into them. So you can't specify any particular area that you want to look at mutations in. Truncation libraries are, as I understand it, libraries that are of interest to structural biologists who are looking to find the largest component of their protein that is soluble. And essentially we're going to nibble off chunks of the protein from one or both ends and give you various truncations. Again, these are mostly going to be individual sequence verified genes, so these are not going to be terribly reasonably priced. But they can be very useful if you want that individual soluble piece of the protein that you're looking at. Another library that is very popular with some antibody screening companies is this antibody library. This is a type of site scanning library where the diversity is focused on the CDRs of a particular antibody. And so you may have, instead of individual mutations that you're combining, you may have a series of 5 to 8 to 10 amino acids all in a row, all in the CDR combining regions, that you're going to diversify in a specified pattern. So I have a couple of specific application examples here. This is one that we made for Genencore, now a division of Danisco. This is a combinatorial library. We make quite a lot of combinatorial libraries for Genencore. They use this one to improve purification yields on Bowman-Burke protease inhibitors. And now you know as much as I do about this particular paper. I don't really know what a Bowman-Burke protease inhibitor is, but the point of this is that you can use our libraries to improve specific properties of proteins of interest, in this case enzymatic proteins. We've made quite a large number of libraries for Atomab. These are the antibody libraries where the diversity is focused on the CDR combining regions. The main point is that we can deliver relatively large diversity, 10 to the 10th, in a focused way, and provide extremely useful libraries for you to do things like antibody screening. And I know that Atomab is extremely happy with these libraries that we made for them, and they've come back repeatedly for more and more of these. Now, this is an example from, we're not allowed to say the name of the company, a large pharma antibody scientist who ordered a library with, again, focused diversity, and they specified in this case the type of distribution that they wanted in the positions 3 through 7 that they were interested in. They then sequenced a bunch of these, and this shows, again, the distribution that they found, and it in fact gave them exactly what they're looking for, so they were very happy. And you can also see that while there were a few, these were supposed to be fixed positions here at the ends, you can see that there are a few non-templated mutations that were thrown in. These were created by truncation mutants in this particular library. but it did not throw them off enough so that it created problems for their screening. So again, for antibody libraries, we can generate you pretty much exactly the kind of diversity that you specify and get you something that's really going to be useful for whatever your project is. So the process that we're going to go through if you decide you're interested in a library runs essentially something like this. Is if you have a protein sequence but you don't have a gene for that, the first thing to do is to create and perhaps optimize for the expression host, and then test a specific gene. And then based on that gene, design libraries that are going to address the specific interests, the specific issues that you're interested in. If you already have a gene with a DNA sequence and you know that it works well, we can then take off and right away start doing the library design based on that. There may be, as I've said before, possible minor alterations that we're going to have to ask you for, but we'll always disclose that in advance of anything that we actually do in physical DNA. If, on the other hand, you have a DNA sequence, a gene sequence, but there are issues, then we can think about trying to redesign that gene, going back to this create, optimize, and testing process before you start actually doing the libraries. One of the things that many customers are interested in is what is the turnaround time for a library project. And typically we're going to try to get these done in a relatively short amount of time. Our goal is to typically get these out to you, depending on the complexity, on the order of 3 weeks, 3 to 4 weeks. Now, for some very complex projects, it may take a little longer than that, but that is always our goal, to get these things turned around as quickly as possible. We're developing a new component of our website for submitting and specifying libraries. I'm not going to show you that because it's actually still under development, although it's going to be deployed either last Friday or sometime this week. The goal here is for you to submit a gene sequence with whatever restriction sites you might need for that. Secondly, submit the vector sequence that you or we, depending on the project, are going to clone this into, and that's only if this is something that is going to be used for your specific project. Establish a reading frame so that we can make sure that whatever this submitted gene sequence is going to be in frame with whatever else you have present in the vector. Enter the diversity that you want to specify for each position that you want to vary, and that might be 1 position per gene, it might be 20 positions in a single gene, and that's strictly up to you, and you can just actually specify that on the website. That then gives you a confirmation report right away so you can see that it's been specified correctly, and then we're going to take a look at that and then get in touch with you either with a quote directly or perhaps with questions about your project so that we can make sure that we understand exactly what it is that you want. Now, I've discussed these briefly, the delivery options for the libraries. Many customers prefer delivery of a linear double-stranded DNA fragment. The reason is because that way they can generate more of their library potentially, and it's going to give you the maximum possible diversity. As soon as the library starts to pass through E. coli, that is going to start limiting the diversity because no matter how carefully you design them, there's always going to be some issue around toxicities. We can deliver this as a sequence-verified fragment, with the sequence verified only on that linear DNA. Or we can clone this and sequence colonies picked from that ligation to give you a better sense of how individual elements of the library look like. We can deliver these as ligation products in your vector or our vector. Of course, it makes much more sense if we're going to do this to do it into your vector. We can deliver as pools of clones, and that's best for a small library. And of course, you always want to keep in mind that when we're doing this, that's going to start decreasing the diversity that you receive. And of course, we can deliver picked sequence-verified clones, at least for libraries with smaller diversities. So that's individual site libraries in particular. Of course, as soon as we start delivering individual sequence-verified genes, the costs do start to climb because there's much more sequencing involved in that sort of project. So thank you for listening. Sorry that I went a couple of minutes over. This is my contact information, so if you have specific library questions, you're going to want to get in touch with me at these numbers and or by email. And even if I am not available at the moment you call, I'll be happy to give you a call back so we can discuss these things on the phone. And now I think we're setting up to take some questions that you have, and I'll perhaps roll back to different slides to answer them as well I can.
So the first question that was asked is, what is the highest actual diversity you can deliver?
The highest that I know about is on the order of 10 to the 13th. Now, let me think about this for a moment. That's the biggest number that I can come up with off the top of my head. It's possible that we could deliver somewhat larger than that, but of course the issue really starts to become how much DNA are we going to be able to deliver for you, and that then relates to how long is that piece of DNA. So if you have a library where you're specifying 2 KB as your library length, the diversity that we're going to be able to deliver just based on the amount of DNA, the physical mass of DNA that we can deliver, starts to be limited.
The next question that I thought was interesting is, what's the percent of stop codons in a random library? And can you design it so that it doesn't have the stop codons?
So if you're talking about a random library that is generated by PCR, the number of stop codons that are generated by the substitutions is going to be relatively low, but we are completely powerless to alter that because we have absolutely no way to direct what mutations are created during that PCR process. And as I said, in that situation, I've been told anyway that the bulk, and that's 80 to 90% of the mutations, are changes in the base, not deletions, not indels. And the indels, of course, are what are going to really generate a lot of stop codons for you. Now, the other type of diversity that you can specify would, for example, be an NNN or an NNK. And in an NNK library, 1 out of the 32 codons specified is a stop codon. That's the TAG stop codon. If you ordered an NNN library, in that situation, of course, 3 out of the 64 codons is going to be a stop codon. I hope that addresses your question.
Before we go on with questions, I just want to, real quick, because I know some people need to get on to their next thing, I want to thank everybody for attending. We'll keep answering questions for a while here, but if you need to move on, you can. And I just want to let everybody know I sent out a link to where you can watch the webinar again. It's going to be posted on YouTube. It takes us about 24 hours to get it formatted correctly and up on YouTube, so give us a little bit of time. But we'll get that up for you. Okay, the next question is, what is the typical amount of DNA you can provide for a random library?
So our typical shipment is specified at around 1 microgram. If you need a larger amount of DNA, we can discuss how much you need and what the cost for that is going to be. But typically our shipments are specified as a microgram. Now, when we specify that, one of the things we at DNA try to do is always over-deliver. And so if we specify 1 microgram, you're typically going to get upwards of 1 microgram. In some cases, I know that the lab has shipped up to 5 micrograms when we've specified 1 microgram. But so we're typically going to specify a single microgram. Now, if you say, well, I need 10 milligrams of my library, we're probably not going to be able to deliver that. But I'm always happy to discuss with you the specific needs you have and what we can at least try to do to get you that.
Great. And another related question is, what's the QC provided that shows library quality?
So that is going to be specified in advance, and really our favorite response here at DNA 2.0 to virtually any question is, it depends. So if you specify that you want delivery of a double-stranded DNA product and you want us to simply verify that by sequencing the actual double-stranded DNA product, that is the least expensive form of QC. And all we're going to do is sequence the piece of DNA itself. And since there are likely going to be non-templated mutations in addition to the, say, if it was an NNK library, the specific NNK position, that sequencing would show you a mixed bases over the NNK part, and then the rest of the product would look relatively correct, although there'd be a low background of other peaks. However, if you then, and we can do this, ligate and transform and pick, say, 96 clones, we can then provide the sequence data from all that 96 clones, and you can see what the actual both diversity is over across that NNK position or NNN position or whatever you've specified, as well as whatever non-templated mutations have popped up. Of course, that level of QC where we're ligating, picking, and sequencing 96 clones is going to be an extra line item on the quote, and we'll disclose those costs completely in advance, and you can decide whether that's an important thing for you or not. So, but we typically are going to do things like ligate, pick, and sequence somewhere between 50 and 100 clones to try to figure out what the quality of the library is for you.
Next question. How could you characterize the hotspot of protein idea in view of your experience?
I'm not sure I understand the question.
You know, I'm thinking they're trying to determine how do you know where the most important area of the protein is to actually look at for the library.
Okay, so this is a question that you can try to address using an alanine scanning library. But I'm going to start making stuff up here. Let's say that you have a protein which is 400 amino acids in length, and you have no idea what's going to be the most useful area to look at in a library. And you also are on a rather limited budget, and you don't want to say, okay, I want all 400 of the amino acids done in an alanine scanning library, because that's 400 genes, and that's going to start to be a little expensive. So you might then try to reason by looking at any homologs where there is structural information to ask, where are the active sites of either closely or distantly related proteins, and what residues on those relatives of my protein contribute to the active site and or the structure that directly supports the active site. And those might be areas where it would be useful to then do a smaller alanine scanning library, perhaps with anywhere, and I'm again making stuff up, 30 to 100 different residues where you would substitute alanine and ask, what effect does this have on my protein? Based on that data, you can then think about, okay, if you find, say, 5 or 10 residues that directly and immediately knock out the function of your protein when you substitute in an alanine, you can then go back and say, okay, now I'm going to take those 10 residues and I'm going to do an NNK, individual single NNK substitution into those 10 positions, and then screen those 10 libraries to say what kind of substitutions in those 10 positions improve the function of my protein, even if only a little bit. After you have that data, you can then think about, okay, so now I have, let's say in those 10 positions, a total of 5 to 10 substitutions that make small improvements in the function of my protein. Now I want to combine those substitutions into a single library, where I'm looking at a combinatorial site substitution library, and you can then say, okay, now if I screen this, will I get larger improvements in the function of my library, my protein, my gene?
So another interesting question, kind of broad, but I still think a lot of people find this relevant. In your experience when designing these libraries, what is actually the most time-consuming part and the hardest to get right?
The hardest to get right is specifying the codons for the diversity positions when you as the customer say, okay, for position X, I want the following amino acids, and then someone would specify, say, 15 different amino acids or codons that they want substituted in. And they say, I want this expressed in E. coli. So then what we're typically going to try to do is pick out individual codons that are best for E. coli for all of those substitutions, those 15 substitutions in that individual spot. If it's a single site library, that's not that difficult to do. But if you've specified it's an antibody library, say, and there are 8 positions in a row, and each of those 8 positions has different diversity specified, and we have to come up with codons for all of that, then we have to design oligonucleotides to encode that. And because oligonucleotides are synthesized 3' to 5', we have to make sure that we do that reverse complementation correctly. And that can be a point at which it's very easy to get confused. And we are right now, in fact, developing new tools to automate many of these steps so that neither you nor we will make mistakes. One of the things that we're trying to do with our new tools is develop reports that will meaningfully report to you the diversity that's been specified, how it's going to be encoded, and what that library is going to look like. Because our production systems are focused on producing individual sequence-verified fixed genes. It's very easy to report those to customers. For libraries where there's variability that is specifically encoded, it's much more difficult for the software that we currently have running in the lab to do the reporting back to you in a way that's going to be meaningful for you. So we're continually evolving and improving our tools to make things better, both for myself as someone who has to do sales and support for these tools, as well as for you, the customer.
Great. Well, that was our last question, so I'd like to thank everybody for joining us on this webinar. I'd like to remind you all that we have another webinar in exactly 2 weeks on protein GPS and infologs to engineer biological systems, which is quite relevant in what we're talking about today because it's the opposite side of, rather than having a maximum diversity, going to that rational design where you spend most of your time on design and a lot less on screening. And with that, we'll say thank you, and if you have any further questions, please don't hesitate to email Dr. LeVay Young and call us if you need us. Thanks so much.